In the last lecture, we saw how the reactions of glycolysis break down the 6-carbon sugar glucose into 2 3-carbon molecules of pyruvate, and in so doing yield some energy for the cell in the form of a couple of molecules of ATP. But this ATP represents only a tiny fraction, really about 3% of the chemical energy that was stored in the original glucose molecule. There's still an enormous amount of energy still left in the pyruvate, which is the end product of glycolysis. Now, if oxygen is available to a cell, then the pyruvate produced by glycolysis is further oxidized by the reactions of the Krebs cycle, yielding carbon dioxide as an end product. Another way to think about this is that by the time the Krebs cycle finishes with pyruvate, all of the 6 carbon atoms that were found in the glucose molecule we started with end up in 6 molecules of carbon dioxide. And remember that the carbons in glucose are highly reduced, meaning that they carry a lot of energy in their chemical bonds. By contrast, the carbon atom in carbon dioxide is in its most completely oxidized state. And this is because the carbon in CO2 is bonded to 2 oxygen atoms, and these oxygen atoms, being highly electronegative, pull those electrons in towards them, preventing them from holding energy. So what the Krebs cycle is doing for the cell is completing the oxidation of a glucose molecule. In today's lecture, we're going to continue our discussion of cellular respiration by looking at the processes cells use to extract this energy from pyruvate, enabling them to produce actually what are huge amounts of ATP, the ATP that organisms need to power themselves. As was the case with glycolysis, there are many biochemical details involved in the specific ways cells further extract this energy from pyruvate. Some of these details are important to emphasize to understand how the process works, but let me also point out that we don't really need to get too caught up with these details to see the big picture. The most important thing for you to bear in mind as we continue this discussion is what the relative energy levels are of the intermediate molecules and how these changes in energy level contribute to the gaining of energy by the cell. What we want to understand is where ATP comes from in the end. Actually, the way that the cell harvests the remaining energy found in pyruvate begins with a first step in which the pyruvate is partially oxidized to form another molecule called acetyl coenzyme A, or usually just called acetyl-CoA for short. This first step yields quite a bit of energy. It's a highly exergonic reaction, and it also cleaves off one of the 3 carbons that are found in pyruvate. Remember, pyruvate has 3 carbons. Acetyl coenzyme A has only 2 carbons. That 3rd carbon is lost as a molecule of carbon dioxide. Then, the acetyl-CoA enters the Krebs cycle, and the remaining 2 carbon atoms that enter this way become themselves completely oxidized, yielding 2 more molecules of CO2. This CO2 is essentially a waste product of the process. And of course, this also yields a lot more energy that the cell can make use of. Let's first take a closer look at this conversion of pyruvate to acetyl-CoA. This reaction is catalyzed by a huge multi-enzyme complex located on the inside of the mitochondrion. This enzyme complex is collectively known as the pyruvate dehydrogenase complex. Pyruvate dehydrogenase is truly huge. It's made up of about 60 separate protein subunits. Essentially, what this enzyme does is to rearrange the carbons in pyruvate, splitting off one of those which becomes completely oxidized as CO2, and then attaching the remaining 2 carbons, which are something we call an acetyl group, to a molecule called coenzyme A. It's the combination of coenzyme A with this acetyl group, creating acetyl coenzyme A or acetyl-CoA, that prepares these carbons to enter the Krebs cycle. Now, coenzyme A is itself a very complex molecule, and we don't really need to be concerned about the details of its molecular structure. However, the significance of adding coenzyme A to that 2-carbon acetyl group is something we should think about, and it's as follows. The bond formed between coenzyme A and the 2-carbon acetyl group is a very high energy bond. It's similar to the high energy bonds we saw being formed between phosphate groups in ATP. Thus, the addition of coenzyme A to this acetyl group is essentially a way of storing energy along with this 2-carbon molecule. This energy comes from the highly exergonic reaction that's catalyzed by pyruvate dehydrogenase. There's a lot of energy released. Some of that energy is used to attach a coenzyme A to the acetyl group. And like the attachment of a phosphate group, this then is a way for those carbons to carry some energy along with them. Now, the change in energy level that occurs from the conversion of pyruvate to acetyl-CoA is almost as great a drop in energy as what had occurred across the entire span of the glycolytic pathway we looked at last time. Some of this energy, as I said, is captured by the attachment of coenzyme A to the acetyl group. Some of the energy that's released by this reaction also is captured in the form of a couple of electrons that reduce a molecule of NAD+, remember that's one of our electron carriers, creating some NADH, the reduced form. So we've captured some energy as electrons, and we've stored some energy in the bond between coenzyme A and a couple of carbons. Now, once the cell has produced a molecule of coenzyme A in this way, the 2 carbons in this molecule can enter the biochemical pathway that we specifically call the Krebs cycle. And it's here where these 2 carbons eventually become completely oxidized and where whatever energy we can get out of them is extracted. Let me state this another way. What goes into the Krebs cycle is a 2-carbon molecule, which has still has fairly reduced carbons in it. In other words, carbons that still carry a fair amount of energy. What comes out of the Krebs cycle are 2 molecules of CO2 for every molecule of acetyl-CoA that goes in. These molecules of CO2 are the most oxidized form of carbon. So what we're doing is putting in reduced carbon, 2 atoms at a time, into the Krebs cycle, and then using the Krebs cycle to squeeze the energy out of those carbons, sending them out as carbon dioxide as a waste product. Now, I want to look a little bit more closely at the Krebs cycle, and the first question we want to ask about this process is, why do we call it a cycle? That is, why do we call it the Krebs cycle and not something like the Krebs process? Well, the reason is that the intermediate compounds in this process, that is, those compounds that are each catalyzed enzymatically from one to another along the metabolic pathway, regenerate themselves in a cyclic fashion. In glycolysis, for example, we started with a particular substrate, glucose, and we ended up with a particular product, pyruvate. We had glucose, it went to pyruvate. When we ran out of glucose, the reactions of glycolysis didn't run. However, in the Krebs cycle, the final molecule that's produced by the series of enzymatically catalyzed reactions then cycles back into the beginning of the process at the start. When Hans Krebs, in the 1930s, remember he's the scientist for whom this process is named, and other biochemists started to look in detail at the reactions involved in the oxidation of acetyl-CoA, they noticed an unusual pattern. They noticed that none of the 8 intermediate compounds, there are 8 compounds in this cycle, none of these 8 compounds ever got used up in any sense. Krebs's greatest contribution was to understand that the reason that this occurred was because these reactions occurred in a cyclic fashion, with the final product forming a substrate back at the beginning. This means that what actually happens is that we have a constant cycling of these 8 compounds around to each other, and they never run out. Now, this leads us with a little question. If the Krebs cycle is a cycle, how does acetyl coenzyme A ever get into it? Remember, those are the carbons that we want to oxidize. Well, this happens in the first step of the Krebs cycle, in which the 2 carbons that are donated by acetyl-CoA become combined with a 4-carbon compound called oxaloacetate. The acetyl group and the oxaloacetate combine to form a 6-carbon molecule called citrate. Now, this is an energetically disfavorable reaction. It's an endergonic reaction, the combination of this 2-carbon molecule with a 4-carbon molecule. This shouldn't surprise us. We're actually making a more complex molecule out of 2 simpler ones, so we would expect it would take some energy. The energy that's used to drive this reaction, in other words, the energy that's used to wedge those 2 carbons into this cycle, is the energy that has been stored in the bond that was formed with coenzyme A. That's the significance of that bond at this point. When this 2-carbon acetyl-CoA combines with the oxaloacetate, the coenzyme A itself is cleaved off, much like a phosphate group would be cleaved off, and it's recycled and used again. What we've done is now taken that 4-carbon oxaloacetate molecule, added 2 carbons to it, but also stepped up its energy level to quite a greater height. Now, there are 2 other things that are worth pointing out about this step. Or I should say one other thing that's worth pointing out about this step. And that is that this coenzyme A actually is used in other parts of the cycle as well. It's used in another reaction. So coenzyme A is actually serving a special function as an energy storage compound that's combining with some of the reactants of this cycle, and in so doing storing energy that will drive some of the reactions that need a little help to get pushed along. Now, once we have citrate formed by the combination of the 4-carbon oxaloacetate and the 2-carbon acetyl group that's entered the cycle, then the remaining reactions of the cycle more or less all run energetically downhill. Now, in a couple of these steps, as you might imagine, one of the things that happens is that we lose one of the carbon molecules. In other words, we have a 6-carbon citrate after this reaction, but as the reactions continue through the 8 steps and cycle back around to oxaloacetate, we end up with a 4-carbon molecule. So along the way, 2 carbons are lost somehow, and those 2 carbons are lost, as I said, in the form of carbon dioxide. One way to think about this, I should point out, is that it's as though the Krebs cycle is sort of like a biochemical furnace. And what we're doing with this Krebs cycle is constantly shoveling in these 2-carbon acetyl molecules, these molecules that have a lot of energy, the carbons in them that have a lot of energy, and burning them up as the cycle progresses around. Now, if we consider just the Krebs cycle itself, once we've got that citrate molecule formed, as I said, the entire process essentially runs energetically downhill. This is different than what we saw last time with glycolysis. Remember, we had to invest some energy in the first steps of glycolysis before we could get any energy out. This isn't the case. Once we've got citrate from the formation of from the joining of a 4-carbon and 2-carbon molecule, everything else is more or less downhill. And as these reactions run downhill energetically, not only do we lose a couple of carbons along the way, but we also capture some energy in a couple of ways. Specifically, for every molecule of acetyl-CoA that goes into the Krebs cycle, we get 3 molecules of NAD+ being reduced to NADH, and a molecule of that other electron carrier, FAD, being reduced, that means picking up electrons, to become FADH2. In other words, what we've done with the Krebs cycle in part is capture some of the energy that's being lost as we step down the reactions in the form of electrons. We also produce 1 molecule of ATP, just 1. For every acetyl coenzyme A that enters the reaction, we get 1 molecule of ATP out. Actually, what that means, for every glucose molecule, we started out at the very beginning, because that glucose molecule will split into 2 3-carbon molecules, 2 pyruvates, and thus produce 2 acetyl coenzyme A's. What that means is we actually get, per glucose, 2 molecules of ATP out. Actually, the way ATP is generated in the Krebs cycle is a little bit more complicated than we saw before, and it's not an important detail, but it's kind of interesting because it shows you really how complicated some of this biochemistry can be. The enzymatic step that creates ATP itself actually has another shunt involved, because the substrate level phosphorylation that occurs, remember that's the phosphorylation that will occur when the energy of an exergonic reaction is directly coupled to adding a phosphate to ATP. Actually, this phosphate is added not to ATP, but to a related molecule called GTP. That stands for guanine triphosphate. And guanine, you'll remember, is just another base that we saw when we looked at nucleotides some time ago. Well, what happens is that this guanine diphosphate picks up the extra phosphate. It becomes GTP, guanine triphosphate, and then immediately the guanine triphosphate donates its phosphate group to an ADP, and we have ATP. What's the point? This is just the way these reactions have evolved. A little more complicated than most students of biochemistry would like, but that's what you have. Well, as I said, as the Krebs cycle proceeds, we've lost 2 additional carbon atoms along the way as CO2. And these carbon atoms lost as CO2 are really something you should think of as a waste product of the Krebs cycle. When we include the 1 carbon that we lost already in the formation of acetyl coenzyme A, originally, before we even jumped into the Krebs cycle, what it means is that for each round of the Krebs cycle, for each pyruvate molecule that we've started that had 3 carbons, we have now generated 3 molecules of CO2. And if we double that, because we get 2 pyruvates for every glucose that we started with at the very beginning, what it means is all 6 of those carbons that we first found in that glucose molecule have now found themselves in the highly oxidized, low-energy form of 6 CO2 molecules. Now, the 4-carbon compound, that's the last compound in the Krebs cycle, that's the oxaloacetate, is also a fairly low-energy compound. In other words, if we're just graphing the steps of the Krebs cycle in a graph of energy over time, we would start out with citrate at the top, and we would start out at oxaloacetate at the bottom. And I just want to remind you that what then happens is that the energy what happens is that this oxaloacetate, when it combines with a molecule of acetyl coenzyme A to regenerate citrate, also is getting a lot of energy. So this combination of oxaloacetate and acetyl coenzyme A essentially steps the whole reaction process back up to the top. So from an energetic point of view, it's as though the Krebs cycle is running down a series of steps, and when it gets to the very bottom, it gets bumped right back up again by the combination of oxaloacetate with acetyl coenzyme A. Well, okay, that's the Krebs cycle. Now, what I want to do is to just stop for a minute and ask again what the cell has gotten out of all of this. We've now completely oxidized 1 molecule of glucose through the reactions of the Krebs cycle, through the actions of glycolysis, then by the oxidation of pyruvate to acetyl coenzyme A, and then finally through the reactions of the Krebs cycle. Now, we don't really have to keep a close tally of what we get out of it in order to understand the process, but I think it's interesting to just sort of see what we've really gotten out of it, just to get us an idea of where we are in terms of gaining energy. Remember, we started way back at the beginning of glycolysis by the cell having to spend 2 molecules of ATP just to get the process started. Then we got 4 molecules of ATP out, so we had a net gain of 2 molecules of ATP from glycolysis. We don't get any ATP when pyruvate is converted to acetyl coenzyme A, but for every 2 coenzyme A's that enter the Krebs cycle, we get out another 2 ATPs. So at the end of the day, when we take that glucose all the way from its most highly reduced form to the most highly oxidized form in terms of carbon dioxide, really all we've gotten out of this is 4 molecules of ATP. That's not really a lot. It's only about 6% of the total free energy that was originally stored in the glucose molecule. On the other hand, the cell has captured a lot of energy in another form, remember. This is in the form of those electrons that have been picked up by the electron carrier molecules NAD and FAD. And we've actually picked up quite a bit of energy that way. For each molecule of glucose that's oxidized entirely, 2 molecules of reduced NADH were produced during glycolysis. 2 more molecules of NADH were produced during the oxidation of pyruvate to acetyl coenzyme A. And then another 6 molecules of NADH and 2 molecules of FADH2 were produced during the Krebs cycle. Now, if we just sort of figure out what the bottom line is here, and we don't worry about the differences between NAD and FAD for the moment, what this means is that a total of 12 of these electron carrier molecules have been reduced by capturing electrons from the oxidation of carbons originally found in glucose. And these stored electrons represent, it turns out, really a very large amount of energy. Unfortunately, whereas the energy that's stored in the bonds between phosphate groups of ATP, and we've gotten a little bit of ATP out, whereas that energy is immediately usable by the cell because we know that ATP can donate that energy to various processes and reaction. The energy that's stored in these electrons that have been captured by electron carriers is not immediately usable. In fact, it's not useful at all because these electron carriers don't have a way to donate that energy to any other useful process that it wants to power. Instead, what has to happen is that the energy stored in those electrons has to be transferred to some other process, a process that will turn that relatively unusable energy into the usable energy form of ATP. And this is where the electron transport chain comes into play, that third process that I began with. This is where the real energy payoff occurs from oxidizing glucose to carbon dioxide. Well, the electron transport chain is so named because it involves a series of linked redox reactions. Redox reactions that accept electrons originally from the reduced electron carriers, NADH and FADH2, and then pass these electrons from one compound to another, gradually stepping the energy level of the electrons down. And in doing so, releasing this energy in a gradual, controlled fashion that can be used to make ATP. Now, in eukaryotic cells, the molecules associated with the electron transport chain are physically embedded in an internal membrane found in mitochondria. In prokaryotes, I should add, which don't have mitochondria because they don't have organelles at all, these molecules associated with the electron transport chain are actually embedded in the cell membrane itself. Now, one reason this physical arrangement is important is that by being embedded in the cell membrane, the molecules of the electron transport chain can be held tightly in close physical proximity in a way that facilitates their ability to pass electrons from one to another through this series of redox reactions. As we'll see later, there's another very important reason that these molecules are embedded in a membrane, which has to do with the mechanism by which they actually produce ATP. Now, most of the components of the electron transport chain are kinds of proteins that are specialized for accepting and passing on electrons in redox reactions. The specific names of these components don't really matter to us. But what is important is the fact that each compound in the chain is only slightly more electronegative than the previous one. This means that once an electron has entered the chain, it will be pulled energetically down the chain, from molecule to molecule, because the next molecule, being more electronegative in the chain, is going to draw that electron towards it. Eventually, at the very bottom of the electron transport chain, bottom energetically speaking, the electrons are finally passed on to oxygen, which is the ultimate electron receptors. Now, the reduced electron carriers, those are the NADHs and FADH2s that we've been talking about, that the cells produce during cellular respiration, interact with molecules in the electron transport chain that are on the high end of the chain, energetically speaking. That is, they interact with compounds that are the least electronegative compounds in the chain. When they do so, these electron carrier molecules, the reduced ones that we've been talking about, will pass their electrons on. So this is how NADH and FADH2 can lose their electrons and have those electrons do something else that's useful energetically for the cell. Actually, another detail that's kind of interesting but not really key to remember is that the electrons that are donated by NADH enter the electron transport chain in a slightly different place along the chain than the electrons that are donated by FADH2. And actually, the electrons that are donated by FADH2 enter the chain at a slightly lower energy level. So when we think about what we're going to get out energetically from these electron carriers, the NADH is actually giving us a little bit more because it's allowing those electrons to step down further on the chain, energetically speaking. As I said, at the very end of the electron transport chain, the electrons that have been moving down this chain through this series of redox reactions eventually end up associated with oxygen. Oxygen is a very satisfactory final resting place for these electrons for a number of reasons. First, as I said, oxygen is extremely electronegative. It's an electron hog. It's the ultimate acceptor of electrons. So what this means is that actually oxygen is really pulling these electrons towards it inexorably along the chain. As electrons are moving from one compound to another along the chain, they're ultimately being pulled in by oxygen, leaving gaps for electrons higher up, energetically speaking, on the chain, which pull yet more electrons downward. Okay? In fact, you might really think of oxygen as driving the movement of electrons in the electron transport chain. Now, the second reason oxygen is a good place for these electrons to end up is that, in most cases, oxygen is relatively abundant. So there's really no need to worry about running out of oxygen as long as we're in an aerobic environment. There's always going to be more oxygen to take the electrons. Now, the third reason that oxygen is a good place for these electrons to end up is that when it receives these electrons, it quickly combines with hydrogen ions in the medium, producing water, H2O. And this shows us where the water comes from in that equation we talked about a couple of lectures ago, the equation for burning glucose. We said that the products of this reaction were CO2, and we've seen where that comes from, and water, H2O. And this is where the water comes from. So, in a sense, water is another one of the waste products of cellular respiration. And water is actually a pretty good waste product to produce because it really doesn't provide any difficult disposal problems for organisms. Now, you might ask why we go through this whole electron transport chain. What we're trying to do is to get the energy out of these electrons. Why don't we just give them to oxygen? Well, it's for the same reason that we wanted to step down the energy levels of compounds in glycolysis and in the Krebs cycle. We want to release this energy gradually, so that the cell can make use of it. Because if it's released too rapidly, the cell just can't simply manage it. Well, now what the cell has accomplished at the end of the electron transport chain is really the complete oxidation of the electrons that the cell had originally captured from the respiration of glucose. The electron carrier molecules that donated these electrons are now in their oxidized form, and so they can be recycled back to the Krebs cycle or to glycolysis to pick up more electrons and keep those metabolic pathways going. But the most important thing that's happened is that the energy that was stored in these captured electrons has been released in a way that can do work for the cell. And the question then is, what exactly is the nature of this work? We know what we want to get out of this. We want to get out the production of more ATP. I just told you that the electron transport chain gradually steps down the energy level of captured electrons through this series of redox reactions, releasing energy gradually. And we know that we want this gradual release of energy to somehow be coupled with the production of ATP, which in fact does happen. But the question is, what connects the two? How is the energy released by these redox reactions somehow captured in a way that produces ATP? Well, the process by which this occurs is what I referred to earlier as oxidative phosphorylation. For many years after the details of how glycolysis works and how the Krebs cycles works, for many years after these details were well understood, the connection between the oxidation of electrons by the electron transport chain and the production of ATP remained a complete mystery. The reason it remained a mystery is because the fundamental mechanism for phosphorylating ADP to produce ATP, that is, the mechanism of substrate level phosphorylation, had already been understood. And scientists thought that somehow a coupling enzyme must also be involved in the connection between the electron transport chain's activity and the production of ATP. It turned out that that wasn't the case at all. What does happen will be the subject of the next lecture.